great pleasure to be here in this beautiful city and this exciting conference and talk about why we need to sequence our genomes and why they're important for the clinical applications. So let me start with the outline of my talk, and what I'm going to cover is why it's so important to sequence our genomes. Can we do it cost effectively? Can we achieve accuracy that is needed? And that's a very, very important, genome is so big without high accuracy in clinical applications, it'll be very difficult. And then some proofs that the whole genome sequencing is usable in the newborn diseases, PGD, prenatal diagnosis, and cancer. And at the end, some vision about genome healthcare. So let me remind you, why is genome so fundamental for to know our genomes for us and for our family members? And one way to summarize that the genome sequence is the ultimate genetic level. There is no deeper level. Once we know the sequence, that's the genetics forever. There is no any science or any expectation that there is some other deeper genetic level than the genome sequence. So it covers all variants, special family variants accumulated in the last hundreds or more years, and de novo mutations specific for each of us. In addition, the genome is a very important. It's predictive. It has a program. It has this, and it's sort of controlling development and functional adaptive functioning of all our tissues. There is no exception. Every tissue is affected. Every of our aspects of our lives are affected by the genome. So it allows, because it's a program, it allows this early pre-symptomatic baseline test, at a single cell before there is any brain or any other organs developed. So that's a really, really important to keep in mind. What is probably most important, the genome with all the power and all the sort of control, it's an open system. It's designed to sort of do interpretation of the signals from the environment for this adaptive functioning. So it's not a destiny. There are some mutations that it's very difficult to repair, but most of the time we can modify. If we know our genome, if we know weak spots in our genome, we can modify. We can sort of live our lives that are in harmony with our genome instead of against our genome. And there are different ways of doing that, through even different food and the genome-specific food, drugs, cell therapies, gene therapies, and many other ways. We should also keep in mind that because of that open architecture, the signal interpretation, even if we know our genome 100% complete and accurate, the predictions are conditional. They are only under certain conditions, a certain environment. For example, we cannot tell from the genome of a newborn baby whether that kid will have cancer in year 50, because that depends on some stochastic factors like getting somatic mutations and the environmental exposures, in addition to the predisposition that are in that genome. So we just have to keep that in mind, that we need genome, but we need many more. We need other somatic measurements during our lives. So if the genome is so important, how many do we need to sequence? And there are lots of estimates, especially in the sort of this research phase, where we have to understand all these different diseases and their genetic and molecular basis. We have to, and one thing is, we shouldn't really focus only on the disease, even though that's the primary goal. As I mentioned, genome is really affecting all other aspects of our lives. So it's our species program. So we should understand every possible aspect of that program. So we need to sequence millions of genomes for that. Obviously, to develop better drugs, to develop better software once we sequence the genome of a newborn to do proper interpretation. And then, obviously, in the implementation phase, and these two phases overlap, to do genomic medicine and genomic healthcare, we need to sequence billions. There are 7 billion people on Earth today. for personalized healthcare and this personalized way of living. There are many other things like, we can envision personalized learning or personalized parenting. What is really very important, many times we don't take in consideration needs of every child. So if we have to sequence millions and billions of genomes, we have to do that really in an efficient and cost-effective way. So the question is that available today? Can we do that soon? Or maybe that's sort of a task that is very difficult to achieve. So over the last 20 or so years, I was involved in lots of companies, lots of research groups were involved in developing something that is called massively parallel sequencing. And maybe that's one way, definitely, today that can provide such a large data set, to sequence one genome accurately, you need to sequence at least 50 times every base on average. So that's already lots of measurements, and then multiply that with millions or billions of the genomes. So the massively parallel sequencing is, sort of the first proposal on using micron beads was in 1988. At that time, I was in Serbia, so this sort of concept was not invented at Stanford or Harvard. It can come from small countries like Serbia. But the point is, it's a very, very simple approach. You take the genome with all complexity, fragment in billions of short fragments, and then do some proper clonal amplification. What we're doing today with this rolling circle replication on the circular DNA, so that we can have enough DNA for every piece of the genome. So we form something that we call DNA nanoballs, about 500 copies. So they are like a concatemer, but it's in a form of a nanoball. So in a single tube, we can make billions and billions of these amplified pieces of DNA in a simple 15-minute reaction. And on the other side, using already developed technology for making computer chips, photolithography, we create these. We create these patterned arrays, use silicon and photolithography. So we have about 4 billion on a microscope slide, 4 billion sticky spots with hydrophobic areas in between. So we simply take that slide and the tube of DNA nanoballs, and in a self-assembly process, we can create these arrays with 4 billion spots on a slide with 1 DNA nanoball per slide. So using this high density, and these are real nanoarrays, the distance between these is today about 600 nanometers, 0.6 microns. So easy to make, these high density arrays, easy to assay because we can use a small volume of reagents because they're packed. And what is the most important, it's easy to image because, again, we have to do all of these billions and billions of measurements. And the reason why is this so easy to measure, because we have this perfect grid where DNA is nicely aligned, so we can align the pixels in the cameras that we use to image so that only one pixel is sufficient to measure one DNA spot. Usually if the DNA is not in a perfect grid, but in some random arrays, like in several other platforms, you need 16 pixels or so. So that's much less efficient. So the instrument can sequence 16 times less genomes. So using machines like that, we have, with this really high precision alignment with a nanometer level, 30 frames a second, we are sequencing today in one run 18 genomes with 50x coverage. With just 24 instruments, we can sequence 1,000 genomes per month today. We already did about 20,000 such high-quality genomes, and this is great. Think about, a few years ago there was only a few genomes sequenced. Now, one small company was able to sequence 20,000 high-quality genomes. But remember my slide that we need to sequence millions and billions eventually. So this type of instruments that are available today on the market, or our instruments, even though with enormous capacity compared to the past, they're not enough. This is not enough. So we need much better instruments. What we're doing now, we're developing a new instrument that has 30 times higher throughput, 30 times more data per day. So this is, in one step, we're trying to achieve something that is really, really, a big jump forward, 30-fold. It's not 30%, it's 30-fold. So, and the reason that we can do that, there are new cameras developed using this computer technology called CMOS cameras, so that they have much higher speed, more frames per second, more pixels. So by using those cameras and integrating our fluidics and our software, we, for the first time, I think we'll be able to sequence millions of genomes per year with a reasonable number, like 50 to 100 of these instruments, and achieve cost that everybody's hoping for, $1,000 genome. And one way is actually to think maybe for the first time with these instruments, we can do whole genome for the cancer with even 1000x to discover these rare variants. So in addition to this, enormous progress in the capacity of sequencing that is really coming, these new instruments are, like prototypes coming in next few months. So next year will be an exciting year where these types of instruments will be start to use them. Is the quality. So in addition to high throughput, we cannot cut corners and have low quality because genome is so big, 6 billion bases, any small error will accumulate and will be high error rate. So this is one way that we measure our quality, and in this case is about which % of bases we can call both mom and dad allele with a high confidence with a small error rate. And if we focus on the coding part, up to 99% of every base in the coding region, we can call it this high quality, where there is only about 2,000 false positive variants per genome. And we measure that. We sequence the genome twice, independently from the same person, and there is only 4,000 discordances, meaning each of them has about 2,000 errors. So this is a good start for doing whole genomes routinely with a quality that, at least for the research, this is very good. But obviously, we would like that this 2,000 becomes 0, only a few errors, and the completeness to get to 100%, and we're working toward that. So I'll show you a couple of slides on a study that we did in collaboration with Han Brunner's lab on using this type of whole genome sequencing to try to figure out mutations that are causing intellectual disability in kids. So the collaboration was designed in a way that, because they did a study and published a study on 100 trios. They had 100 kids with disease and their parents. So they did exome sequencing, and they found 16. They explained 16 disease. They found causative variant in 16 kids out of 100. So they sent us 50 of these kids that are negative by their exome sequencing. So we did parents and the kid 150 genomes with a high quality 80x coverage. And this is one way to summarize the result. So what they did here, after, and all of this, it's really important, all of these variants, and from their prior study, over 90% of the causative variants are de novo mutations that don't exist in parents, they're present only in the kid. So they looked de novo mutations in our set and focused only, for the moment, they just focused on the coding. That we sequenced whole genome, but they said, we'll look only the exome part of your data. So they validated out of all variants that we found as a de novo variant, they validated 74 in these 50 samples. This is the European period, the dot. So 1.48 variants per genome, de novo variants in the coding region per genome. And when you look at other studies, this was we had the highest number of de novo mutations validated per genome relative to the other studies. So this actually demonstrates how this technology is improving. It used to be only half, and then there was better and better. There are a couple of studies in the last couple of years that are getting to this completeness that is really important. Anybody who was doing studies at this time, they would discover only 50% of the causative variant. So this was really good. So our genomes were discovering the largest number of proven, confirmed, validated de novo variants. In addition to that, they found 6 clinically relevant de novo copy number variation in these samples that they couldn't find by the Affymatrix chip. And this is one example. As you can see, father doesn't have any deletion. So the deletion is shown as a red coverage, low coverage. But mother had one spot, and then de novo mutation happened, so the kid has two, probably disrupting both alleles of that important gene. So in a summary, when they looked all these de novo mutations and they looked biological, medical relevance of them, so as I mentioned before, they explained 16%. So they already had, and they, in the set of descendants that are not these 16%, we've explained another 38%. So combined 54, there are some variants that are accurate as a variance. They are validating, medical relevance. So with this, probably this all together, just in the coding, they're still just looking coding, it will be 60% this diagnostic yield. I think everybody can expect at least 20% to be explained with the non-coding. So I think in a year or so, we'll be having results that for the intellectual disability, for the diseases caused with de novo mutations, that we can explain 80 or so percent. It's almost like unbelievable yield. So the message from this, do whole genome sequencing first. Don't do all of these kinds of partial analysis, different diagnoses, spend years and years to try to make diagnoses for these poor kids. Do genome sequencing first, and then if that doesn't explain, use other techniques. So one really interesting sort of conclusion from that study is if explanatory power is so good for the kids that are sick, can we do now that prenatally or in PGD? Can we detect with same accuracy these de novo mutations and prevent having kids with these terrible diseases? That'll be the real way of doing preventive medicine. The problem is to do that in the PGD, there is only a few cells from these embryos. 5 to 10 cells is the best that we have. So we need we need to have a genome that is even higher quality for those applications. So what we need, in addition to our current standard genomes, we need to phase. We need to today there is a consensus. We don't know whether it's mom's or dad's variant, so we have something like this. We need something like this. We have to separate variants that belong to mom and to dad. So that's the first important thing. The other is we need to solve these repeats. There are so many pseudogenes, and so we really don't know when we see some of these variants, are they causative or not. So most likely, what we need de novo assembly of individual genomes. Even though there is a reference, we should ignore the reference. So that's one of the goals that will be good to achieve. We have to remove these remaining errors, but hopefully go down to close to zero, and do all of this on just few cells, 5 to 10 cells. That's what we have. There is no more than that. So can we do it? So we've proposed several years ago a technology called long fragment read technology, which is pretty simple to implement. And the idea is really sort of coming from the information theory. So what we do, we just take, say, 10 cells, isolate DNA in the long fragments, 100 kilobases or longer, and then take all of these fragments and aliquot, just pure aliquoting, in a plate, 384-well plate by a robot, just pipetting. So what is happening in this stochastic separation, that fragments that belong to mom and dad, they go in different wells. So there is this stochastic separation of haplotypes and repeats. So what we do in the plate, we just amplify DNA, fragment down to the final fragments that we need to sequence, ligate a barcode, DNA barcode specific for each well, pull this together and sequence. So it's a few pre-processing steps using robotics, and then everything else is the same type of high throughput sequencing. So using that, we published a paper last year in Nature demonstrating that this can provide high accurate genomes, and for the first time, we're able to show that we can do high efficiency phasing or haplotyping. 97% of heterozygote SNPs were assigned to parents. Very long haplotypes or full chromosome haplotyping when you have a genotyping of one parent. And as I mentioned, the interesting benefit of this, when you're doing this separation and phasing, if there is an error, it doesn't fit. It's sort of like, how can this read that read G, some reads belong to mom, some to dad. It cannot be like that because we now separately sequencing mom and dad. So we've proven that we can reduce error rates from 2,000 or so to about 300 per genome. And at the end, all of this can be done on just a few cells, 10 cells. But it's important to realize it's not one cell. So we need sufficient number, 5, 10, 15, 20 if we have, because they sort of control each other. Anything that is not reproducibly found in enough cells, it's most like an error. So we wanted to test, can we do this as a sort of the ultimate PGD? Can we sequence 5 cells to 10 cells from the 5-day embryos, from in vitro fertilization embryos, with high accuracy so we can detect de novo mutations in these embryos, what was never done before, because usually people will just look for those variants that parents have, for the inherited mutations. So we organized a study where we took a couple of embryos from a same couple, and from one embryo we did 2 biopsies from the same embryos. We wanted to prove that we can sequence with high reproducibility 2 biopsies. And here, as you can see, we have 90%, 91% concordance if we don't do this phasing, if we don't do this haplotyping. As soon as we do that, it goes to 98%. So that was a really good thing. So the variance shared between these 2 biopsies are at the high level. We know that we don't have 100% sensitivity, so that's why it's only 98%. But it's a good proof. So then the question was, can we detect de novo mutation with a acceptable, tolerable number of false positives? So always balance between sensitivity and specificity. So this table shows that that's possible, that with this type of data, having 10 cells is really possible. So what we did is 2 million variants detected in one of these biopsies. We said, okay, we have parents, so we're chasing now false positives in this column and de novo mutation in this column. So we say if something is a false positive, it shouldn't be found in parents or any other embryos or in other published genomes. So when you remove all of that, we're left with about 900 variants that we cannot explain. They appear as false positives. That's a pretty large number when you're chasing the 100 or so de novo mutations. So there'll be more errors than de novo mutations. But what we discovered, because we have this 384 valves, and because we have 10 cells, we also denatured DNA, the actual variant should be, reads for that variant should be found in 15 to 20 valves. Should be in many valves represented. If there is an artifact or other things that are, as a sort of, without looking at the valves, we call a variant, but they shouldn't be in so many valves. And that's exactly what is happening. If we ask how many variants are in 6 or more valves, only 25 out of this 900. Obviously, we then check the sensitivity and say how many of the true variants that are shared with parents have these 6 or more valves, over 90% or about 90%. So in a way, we can improve sensitivity, we can improve specificity 50-fold, reduce false positive to 25. Most likely, some of these or half of these are still not false positive. We just don't have evidence in the other genomes, and lose only 10% of sensitivity. So as a final result, we think we can detect about 80% de novo mutations in these small biopsies, microbiopsies, with almost non-existent false positives. And that will be for the first time that we can really avoid implementing embryos with de novo mutations that are causing a couple of % of newborns this intellectual disability and other diseases are caused by de novo mutations. So that's a sort of a first demonstration, and obviously lots of clinical studies will be done in the next couple of years and implement that in a real life. So the other application of this technology, again, with a small number of cells, would be circulating tumor cells, where it's like a liquid biopsy, where from non-invasively from 10 ml of blood, people have different technologies to collect about 10 cells. So what we did here in collaboration with UCSF, on a patient, breast cancer patient, they isolated several preps of 10 circulating tumor cells. So we applied the same technology, and when you do blood cells, you see the normal copy number variation. This is X chromosome 1 copy in this male, and all others are same. But when we did 3 of these liquid biopsies, we see reproducibly changes in this tumor, circulating tumor cells. As expected, there are chromosomal changes. So this is a good sign that this type of technology and this collected circulating tumor cells can be analyzed and give us a clue what are the genetic changes that are causing that cancer to grow. As a summary, what we found, and in addition to copy number, we also have obviously the single base variation. We found that CDH1 has a frameshift mutation and it's present in this one copy deletion, and BRCA1 is also deleted in one copy. So it's almost like already there is a signature of how is that cancer, what is driving that cancer, so that the best therapies specific for that type of cancer can be selected. So a few slides to close on this, the view of the genomic healthcare, how to live longer, healthier lives using the genomic information. It's obviously a new healthcare, a new approach, and we should really keep in mind that it requires several components. In addition to, obviously, the first component is highly accurate whole genome sequence, but in addition to that, as I mentioned before, we need more measurements from the real, real tissues and organs, and we need that to do regularly, and I call them iomics, individual omics measurements, expression, epigenetics, and so on. But in addition to these measurements, we need to provide that in some personalized electronic health coaching approach, that to enable that people use these reports. And then obviously we need to work on some genome-specific prevention, some drugs and supplements, remedies for that. So the goal is really, or the approach is very simple. Sequence the genome as the ultimate genetic test once, store that in the computer, do these sort of computer-generated reports when they are needed and that are actionable. Because we store all information, we don't need to report the drug response for a person that doesn't use any drugs. That's meaningless for that moment for that person. But that's available whenever doctor has to decide on prescribing a drug. And sort of the idea is that this is individual genome tailored health care, and primarily it's a disease prevention, and then obviously better diagnosis and better treatment. So as a cartoon, to express this, we really need to integrate the genome as the sort of infrastructure, as the fundamental level with these other measurements and other tests, other medical diagnoses that are needed. And more computers have to integrate this and provide these usable reports. Doctors and scientists are unable to integrate all these data sets, so we'll be developing more and more efficient software to do that. So as a summary of why is whole genome sequencing much better approach than any targeted sequencing, obviously today we cannot do a large-scale whole genome sequencing on millions of people, but the idea is, let's get prepared. Let's think about this as something that is coming. Technology is going to enable that. Let's think from the policy point of view or the economic point of view. Why is this important to start preparing and using it? And my way of thinking about this is the first obvious thing is when you do a genome, you get all the variants. So it's a sort of in a cost-effective way. So that's a critical component. So when you do that, you can now flip instead of these sort of qualification, whether it's a family disease or not, for BRCA1, and we miss 50% of cases. You're doing qualification to save on the cost of one gene. So doing that, let's do whole genome because it's for all diseases, and then you don't need to qualify. Just do everybody has it, because obviously everybody of us will have some type of form of disease or drug response and so on. So that's the first thing. It's sort of an effective way to do screening versus ineffective qualification. The second, maybe even more important thing is we heard great presentation yesterday in a keynote presentation about this incomplete penetrance. This is just one example of many other reasons why looking at the single variant as a targeted variant doesn't explain most of the cases. Only, the extremes can be explained because other variants in the genome contextually define the meaning and the expression of that variant. So the genome, because we have all the variants, finally will allow us that. Otherwise, it will be impossible. So by combining, more comprehensive approach, screening approach with better interpretation, and at the end, have the genome being ready for the future development of this field. Obviously, today we cannot explain many things. And also have your genome ready for like urgent care or sort of just available. Imagine today that the doctors are doing surgeries without X-ray. Nobody would do that. It's the same thing, I think, in a couple of years. Nobody will do any type of treatment or disease diagnosis without having genome behind it. Another important aspect is more like a social aspect. So if the genome is like infrastructure, if the genome is the ultimate level that is going to stay forever, nobody says, okay, we'll be sequencing this genome 10 years and later we'll invent something else. There is absolutely any prediction like that. There's no reason not to do whole genome sequencing forever. Suddenly, if that is the infrastructure that the healthcare needs, the policymakers should think about, let's make this as a priority and develop this infrastructure, because if we have that, suddenly we can have more cost-effective approaches in diagnosis. For example, we don't need to develop thousands and thousands of the companion diagnostics for every specific drug. It's a lookup from the genome. We can have more efficient drug development and more efficient clinical trials where every person will have their genome when they enroll in the clinical trial. And obviously the more efficient healthcare, because we should avoid this using treatments that don't work. So it's really the genome should be seen as like an internet infrastructure, so that we can do all of this. So I think I convinced you that this massively parallel sequencing provides the way today, before some other technologies, and including my team, lots of people are working on trying to do single molecule sequencing. But for now, that provides really high throughput, high quality genomes for both reproductive health and genomic healthcare. What we need to do millions of these genomes to provide a knowledge base so that we can do better interpretation, develop better software, implement this e-health, and start this standardization. And obviously, we have to educate general public, especially, and medical professionals, how to use this. Thank you very much.